My name is Lisette Dominique Bujarrey. I am a master's student at North Carolina State University, and I am doing my master in physiology with a certificate in biotechnology. I want to talk to you guys about my favorite picture that I took during these 2 years of my studies at NC State. So even though I took other pictures that didn't make it to this video, other pictures were really nice as well and meant a lot to me, but not as much as this picture because it took a lot of effort, not just from me, but from other scientists to develop it. I interviewed Dr. Robertson and Dr. Johannes. Dr. Johannes is the one who handles the machinery to take these pictures because it's very expensive, and Dr. Robertson is my professor in this class. So let's see what they have to say about this.
So this is a beautiful image, and it reminds me of how far we got along in technology when you compare what was possible about 20 years ago. So it's really fascinating to me what we are able to visualize with these new instruments. The beauty of these micrographs is always striking to me. These images speak at a much deeper level to people than just descriptions. So they make science more accessible and also more memorable to everybody.
I love that picture because to me it is my favorite group of neurons in the brain, and in particular in the rodent brain, mice that I study. And it's a group of neurons called norepinephrine neurons that are really important for a variety of functions in the brain. And they help things like your attention, so your ability to listen to what I'm saying right now depends on those neurons doing what they're supposed to do, right? So if they release too much norepinephrine in your cortex, this part of your brain, then you might be looking out the window. Or if they release too little norepinephrine, then you might be falling asleep as you're trying to listen. And they serve a variety of other functions, and including things like attention and about if I stimulate a certain population of neurons, does that impact the structure and development of the brain in a certain way? And does it in fact impact behavioral and physiological processes in a particular way?
So these norepinephrine neurons are really important to us. Now, even though we're using the mice as a model, it also applies to us humans because norepinephrine neurons are important for pain, attention, and learning. So in class, we injected these neurons with synthetic drugs, and they would turn on or off receptors. And this took a tremendous amount of work from other scientists and from us to even understand it in the class.
And so that's part of what I'm trying to discover as a scientist. And in class, my students and I, we work together on that, and we have this cool tool, a mouse model that allows us to express colorful proteins like green fluorescent protein, that bright green, or red fluorescent protein in those different populations so we can see the different neurons. And we can also express other proteins that give us control over those neurons in living animals. So they can express a receptor, so we can inject an animal with a drug and stimulate that receptor, turn on those neurons, and have remote control essentially over those neurons in living animals. And even in embryos in a developing mother, called a dread receptor. And it's a designer receptor exclusively activated by designer drugs. So that's a mouthful. But human receptors from our brains that were engineered so they no longer respond to the normal ligand that's in the mouse brain, acetylcholine. So they've been mutated so they no longer respond to that chemical, and now they only respond to a synthetic chemical that we can inject into the animal. And so
after all this work, and after we got our little slides with our norepinephrine neurons, we have to take a picture with a microscope, and it wasn't any microscope. This is really expensive. Thus, we cannot use it by ourselves. Dr. Johannes was the one who helped us handle this machinery. The movie is about 60 pictures of the same slides in different directions that is combined. So it's pretty cool.
Oh, it was expensive. The microscope, as you see it here, was about $770,000, which is a lot. So, and we were so lucky of a confocal microscope is that you can optically slice your specimen in very tiny sections without physically cutting it any further. So it's non-invasive, and you can even do, as a result, you can turn around and view at different angles and animate.
So those pictures come from an animal that have 3 genes that have been manipulated by scientists. 3 separate strains of mice were then bred together through multiple generations to create that ultimate animal that gave us that beautiful image. And there's a lot of work. It can take up to a year to develop a single transgenic animal.
There you go. There is the story behind the picture. It's my favorite picture because it took tremendous amount of work to develop it from my part and from other scientists for years. But neurons have a special place in my heart. They are beautiful, as you can see in these images, and their expression and what they do for us in the body is amazing. So if we're able to understand how these pathways work through these receptors and through this technology, such as the microscope and other stuff that we use in the lab, we are able to understand how pain, attention, and learning works. So if we understand how it works, then we can understand how to fix it. So this is why it's so important, and this is why I like it so much. But thank you guys for watching, and I hope you learned something today about neurobiology. I just wanted to say that. Alright. Thank you.